Before a patient ever feels their chest tighten, a microscopic physical collision occurs. Jagged allergen particles, whether from pollen, venom, or food, crash into the outer membrane of a mast cell. These particles lock on to IgE antibodies that are already anchored to specific receptors on the cell's surface, known as Fc epsilon Ri. When an allergen binds to two adjacent IgE molecules, it cross-links them, pulling the receptors together. This clustering acts as a mechanical switch. It reaches inside the cell and activates specific intracellular tyrosine kinases, primarily Lin and Syk. Once activated, these kinases spark a rapid chain reaction, phosphorylating targets down the line until they awaken a crucial enzyme called PLC-gamma. This flowchart tracks the exact sequence. We see the signal move from the allergen and IgE complex down through Lin and Syk, activating PLC-gamma. PLC-gamma acts as biological scissors, cleaving the membrane lipid PIP2 into DAG and IP3. Following the IP3 branch down to the endoplasmic reticulum, it binds to receptors there, forcing the organelle to suddenly dump its stored calcium into the cell. This sudden spike in intracellular calcium is the universal ignition switch for the mast cell. It pushes the cell past the point of no return, launching a highly choreographed two-phase chemical attack. The rising calcium forces internal vesicles to rush to the cell's outer boundary, fusing with the membrane and instantly blowing their contents into surrounding tissue. This is phase A, the immediate preformed granule release. Because these chemicals were already manufactured and stored inside the mast cell, this massive payload is deployed in a matter of seconds. Looking at the left side of this diagram under immediate preformed granule release, we see pathway 1, histamine. Once released, histamine rapidly diffuses and binds to H1 receptors located throughout the body. Following the first arrow down to the lungs, histamine hits the H1 receptors on bronchial smooth muscle. This causes rapid contraction, creating the bronchoconstriction that produces the classic asthma wheeze and sudden anaphylactic bronchospasm. Moving to the middle branch, we track its effect on the vascular system. Histamine forces endothelial cells lining the blood vessels to contract. Microscopic gaps open up, allowing plasma to leak out into the surrounding tissue. This capillary leak drives interstitial edema, hives, swelling of the lips and tongue, and a dangerous drop in blood pressure. At the bottom branch, histamine stimulates sensory nerves, resulting in intense itching, sneezing, and pruritus, while simultaneously commanding local glands to massively increase mucus secretion. Clinically, this cluster of H1 receptor effects is summarized by a simple rule: histamine triggers fast swell and squeeze. This rapid dump of preformed histamine dictates the initial presentation of a severe allergic reaction. It is the chemical driver of the acute, immediate respiratory and circulatory failure that paramedics face upon arrival. Alongside histamine, the mast cell instantly ejects pathway 2, tryptase. This is a preformed serine protease enzyme. Tryptase acts directly on the physical tissue structure. It breaks down the proteins in the epithelial lining of the airway, causing damage that exposes the underlying sensory nerves. Stripping this protective barrier creates hyper-responsive bronchi that twitch and spasm violently. It also worsens local airway edema and sends out biochemical signals to recruit additional immune cells to the damaged site. Furthermore, tryptase activates fibroblasts, the specific cells responsible for laying down structural connective tissue. Over time, this contributes directly to physical airway remodeling. This specific damage pathway explains why chronic asthma progressively worsens. Repeated mast cell degranulation means repeated tryptase exposure, structurally degrading and thickening the airway over years of separate attacks. Because mast cells release this enzyme in such high volumes, we can measure it accurately in the blood. As the central blue box on this chart notes, an elevated tryptase level is the gold standard biomarker to clinically confirm that anaphylaxis has occurred.
Tryptase's role is defined by two primary clinical outcomes: damage and remodeling. While histamine dictates the immediate vascular and respiratory crisis, tryptase sets the stage for long-term tissue destruction and chronic hyperreactivity. The mast cell's attack does not end once the preformed granules are gone. Phase B is the delayed, sustained attack. Taking several minutes to execute, the mast cell begins to manufacture entirely new lipid mediators. It does this by harvesting the phospholipids that make up its own cell membrane. Looking at the upper right quadrant under newly synthesized lipid mediators, we see enzymes stripping lipids from the membrane, converting them into arachidonic acid. Following the left green arrow, arachidonic acid enters the 5-LOX pathway, producing leukotrienes. Specifically, this generates cystenyl leukotrienes, LTC4, LTD4, and LTE4. Biologically, these molecules are some of the strongest bronchoconstrictors in the human body. Looking at the lung icon below this pathway, we see the clinical impact. When these newly synthesized leukotrienes bind to the cis-LT1 receptor in the airways, they drive an intense, smooth muscle contraction that lasts significantly longer than the initial histamine squeeze. They also trigger a massive overproduction of mucus and increase local vascular permeability, forcing the physical walls of the airway to swell inward. The leukotriene pathway is recognized by its clinical hallmark, a long squeeze and mucus. Synthesized continuously after the trigger, these leukotrienes are the exact biological reason an asthma attack can dangerously persist for hours, long after histamine has peaked. Arachidonic acid doesn't all go to the 5-LOX pathway. There is an alternative metabolic route making up pathway 4. In this diagram, if we gray out the 5-LOX side, we can follow arachidonic acid entering the cyclooxygenase, or COX pathway. Here it is converted into PGH2, and finally synthesized into prostaglandin D2, or PGD2. Prostaglandin D2 exerts direct physical pressure on the patient. It actively contracts the bronchial smooth muscle to worsen the airway squeeze, while simultaneously dilating peripheral blood vessels, causing visible flushing and further hypotension. But its most critical function is immunological. PGD2 acts as a powerful Th2 signal amplifier, a chemical beacon specifically designed to recruit eosinophils to the site of the reaction. This influx of eosinophils brings highly toxic granules into the respiratory tract, directly driving the pathology of eosinophilic asthma and chronic allergic airway inflammation. The signature of PGD2 is squeeze and eosinophils. Through this cellular recruitment, PGD2 serves as the biological bridge transitioning a sudden acute allergic response into a deeply entrenched, cellularly driven inflammatory state. Stepping back from the cellular level, these 4 individual pathways combine to dictate the macroscopic clinical timeline of a patient in acute distress. The patient's presentation scales systematically, moving from an immediate wheeze and airway edema in seconds to a prolonged mucus plug and severe squeeze in minutes, to deep tissue inflammation and remodeling over the following hours. The clinical summary flow at the bottom right maps these 4 distinct chemical pathways to the symptoms seen in the field. Highlighting the green box on the left, the acute crisis begins with histamine, generating a fast swell and squeeze. Moving to the yellow box, the newly synthesized leukotrienes follow minutes later, locking in a long squeeze and mucus. Highlighting the remaining 2 boxes, PGD2 amplifies the squeeze and recruits eosinophils, while the preformed tryptase initiates localized damage and remodeling. Bringing more attention to the final gray result box, we see the physiological endpoint of all 4 pathways converging: narrowed airways, severe mucus plugging, and massive edema. The inevitable result is profound respiratory distress. Severe anaphylaxis is never a single point of physiological failure. It is a precisely choreographed cascade of mediators, and internalizing this timeline allows for targeted pharmacological intervention.